All right, seems quiet in here today. It's the rain or something. So a few comments from last time. A couple of you had questions about a couple of things, so I want to just talk over a few points one more time. So a couple of you have questions about viral envelopes. So as we were finishing last time, I said that envelope viruses are a little bit wimpy in the environment. They degrade really easily, they dry out. So a few of you ask, well, what good is it then? Remember, a virus in the environment might have very different properties than inside our body. All right, so inside our body, the envelope is going to be another layer of protection, especially from our immune system. So while it's going to help the virus in the body, outside, maybe it's not as very environmentally tough. It's kind of the opposite for a naked virus, one that doesn't have an envelope. It's going to be kind of tough in the environment, where maybe inside it's going to be more susceptible to some of our aspects of our immune system. Okay, and then remember, we also talked about nucleoside analogs. A little confusion about what a nucleoside analog is. The examples that we gave that are probably a little bit more memorable, like the COVID drug, molnupiravir, it's basically something that looks like a regular nucleoside. So when say a virus incorporates that nucleoside into its whatever, nucleic acid, DNA, RNA, it will do that, but then either it stops replication or transcription, or it just causes so many mutations that eventually that's catastrophic for the virus, and it won't make a viable copy of itself. All right, so basically those nucleoside analog drugs will just stop the viral ability to replicate. Okay. The other just quick comment that I wanted to make. So remember, your next exam is Tuesday. So make sure you got your homework done by class time. A lot of you are rusty, or maybe you have never filled out Scantrons before. So remember, you do need to bubble in your username. So that's, whenever you're logging into Blackboard, for example, that's your username. And that is how I know, I get the results basically emailed to me, and that's how I know who you are. So you've got to bubble that in and not just write your name. And you also need to put the form down. There was about 15 of you. I had 2 different test forms. You have to put down whether you're A or B. Now, the computer figures that out. It just takes me a little bit more messing around. Last year, I got so tired of people forgetting to put their test form on it that I made a question. The first question on the exam was, I have bubbled in the exam form. 2 people said yes when they hadn't, so they got it wrong. So I won't remind you to do that. It's just down at the corner, just A or B. Okay. Okay. So we need to get through the rest of the viruses today, because I'm planning on that being included on the exam on Tuesday. I think that's a pretty doable thing. But I don't want to rush. Stop me if you have questions, and we'll adjust what we get through. All right, so where we left off, kind of thinking about general, almost done talking about general characteristics of viruses. We're looking. We will go into more detail about the nucleic acid as well. But remember, we said that a virus either has DNA, it could have double-stranded DNA, or single-stranded DNA, which of course is something that we don't find in any other kind of cell. So that was kind of mind-blowing when we discovered that. Or it could have RNA, and that can be double-stranded RNA, which again, is kind of unique, or single-stranded RNA. Okay, and it will have one or the other. That nucleic acid can either be circular, like we find in bacteria cells, or linear. And if it's linear, it's either going to be in segments or unsegmented. Okay, so for example, this variant up here, this is a linear genome. You can see these sticks right here. There's 3 different, or 6, it looks like there's 3 pairs here for this particular one, 3 different segments. Coronavirus, if we looked at that genome, would have just one big strand of RNA. Okay, so that would be unsegmented. So all the genes are just in a row. All right, so we see, you know, different things, different viruses. One hallmark is that the gene number is small for viruses. How small is it? Well, if we compare to humans, before we sequenced the human genome, we assumed that there was probably at least 100,000 genes, probably more than that. When we actually went through and sequenced, you know, all the base pairs, we now think that there's more on the order of 25,000 to 40,000 genes. You can't say exactly, like just from the sequence of bases, but we think more in that neighborhood. E. coli, at least a magnitude less, has about 4,000 genes. Compare that to the virus that has the smallest number of genes, and that's hepatitis B virus, only has 4 genes. And that makes sense when you think about what viruses do, right? They are going to enter a cell and basically take over that cell and use everything that the cell has. A good analogy for this, my son, when he came home for fall break a couple of years ago, he brought with him his laptop and a football. And I said, "Where's the rest of your stuff?" And he said, "Oh, no, this is all I brought." He was coming home for 4 days. And I did my mom's side, but then I thought, oh my gosh, he's just like a hepatitis B virus. He knows that at home he has a bed and we provide food for him, and he's got towels, he's got a toothbrush at home, he's got old clothes. He didn't really need to bring anything with him. Everything he needed was already there. He had to do some homework and he wanted to play catch. So those things we didn't have at home. And everything else was provided for him. So again, viruses, they come and stay in the cells, and mostly the cell's going to provide everything they need. So we'll talk about in just a second some of those things that they do need to bring with them. But here's kind of an interesting picture from your book. We have ways to kind of smash open cells and then be able to see that DNA. And so here you can, all these little wavy lines are the partial genome of E. coli. And this one little strand right there, that's some virus. All right, so again, much smaller genomes. What are some of the unique things that viruses need? The information that they need that the cell might not provide? Well, they might need some instructions to make proteins that our cell wouldn't normally make, like for viral spikes. They may need some genes to kind of turn on or off host cell genes, the expression of them. So they might need to regulate the host cell a little bit. And really, most importantly, they may need some genes to replicate their genetic material. All right, so little reminder down here, sometimes I talk about viruses before we talk about genetics, but hopefully you all kind of have this down now. If the virus is a double-stranded DNA virus, then it can do replication just like we do, use the same enzymes, transcription just like we do, use those same enzymes, and translation in the same way. But if it's an RNA virus, say a single-stranded RNA virus, for example, it needs to make RNA from RNA. right? That's not something that we ever do. So it has to bring in its own enzyme, again, to copy its own RNA, and sometimes that's called replicase. Sometimes we call it an RNA-dependent replicase. But that's the idea. Again, we don't do that in our cells. Some viruses, like HIV, is a positive stranded, we'll talk about that in a minute, RNA virus, but it actually doesn't copy RNA from RNA. It makes DNA from RNA and then copies the RNA from DNA. So it uses an enzyme called reverse transcriptase, again, which makes single-stranded and then double-stranded DNA from RNA. And that's something we never do in our cells. So those are just some examples of some gene functions that viruses might need. Before we get into exactly how viruses are going to replicate themselves, I want to just spend a minute thinking about viral classification. There's two different ways, two different schemes that we use to classify viruses. And the one way called the hierarchical classification. I think I spelled that right. So that's kind of the more common way that we have traditionally tried to classify viruses. With this scheme, we're kind of acting like viruses are living. We'll talk about soon whether we can really consider them as living or not. But we try to we act like they're living, and we try to kind of fit them into the taxonomy that we have for living organisms. So basically we have actually updated a little bit, or was looking into this a little bit closer, and I need to update this a little bit, but now there is a phylum that's been described. As of 5 years ago, there wasn't, we didn't have we didn't have class, they just had orders. They had 3 different orders for viruses, and then all viruses are going to fall into a family. And with viruses, you know that it's a family name because that virus name is going to end in viridae. Okay, so for example, here's a family name, Bunyaviridae. Okay, you see that and you know that's a family name. And then all viruses are going to have a genus as well, and they have the suffix virus. All right, so the genus Phlebivirus is in the family Bunyaviridae, and I know those aren't very recognizable. And I didn't write it here, but the species is a little bit more loosey-goosey. With viruses, we usually call the species the disease that the virus causes. Okay, so for the example that I'm showing you down here, Rift Valley fever virus. Okay, Rift Valley fever is the disease, and then we again call that a virus. So that's just an example of the taxonomy with hierarchical classification. It gets a little bit, again, some of those families are not very recognizable, so it gets a little bit cumbersome. Some of them are easy, like herpes viridae, that's a family. Herpes virus is the genus, and then chickenpox virus would be the species. So sometimes it flows a little bit better. How do we decide kind of what class and species and family to put these organisms, or sorry, order, family and genus to put these organisms in? Well, we look at their observable characteristics. What kind of nucleic acid do they have? Is it RNA? Is it DNA? What kind of capsid? Is it helical? Is it icosahedral? Is it complex? Does it have an envelope? Maybe a little bit about how big the virus or how big the capsid is. Now, in addition to those characteristics, we're looking more at molecular characteristics, looking more comparing sequences of, say, the nucleic acid in order to put viruses in groups. Okay, so we're relying a little bit more on that. The other way that's not quite as, it's a little bit more cut and dry, I'll say it, is the Baltimore classification. And this classification of viruses just depends on what kind of nucleic acid the virus has. So there's actually 7 different groups or classes. And this is not to memorize. I don't have these memorized, but for instance, class 1, those are our double-stranded DNA viruses. Class 2, single-stranded DNA. We'll talk a little bit more about the difference between plus sense single-stranded RNA and minus sense. There's our single-stranded RNA viruses that go through a DNA intermediate. Okay, so those are retroviruses. And then there's this other really small group, they're double-stranded DNA viruses, but they don't replicate their DNA. They actually go through an RNA intermediate to make more DNA. So that seems like a lot of extra trouble to go to, but that's, I think hepatitis B virus does that. All right, so 2 different classification systems. David Baltimore, he did a lot of work on AIDS, early 80s. It's been, he's the one that thought up that system. All right, so just to kind of, again, appreciate the complexity a little bit, I thought that I would, this kind of a neat graphic here that just kind of shows you the different ways that we use to put viruses into groups, kind of shows you the different shapes that we see. Again, it's got RNA, DNA, the symmetry of the capsid, whether they've got an envelope, even includes the Baltimore class. So anyway, not anything to memorize, and I don't think this is an exhaustive list of every virus that there is, but it's kind of interesting, helps you kind of see what kind of things we use to classify. All right, so viral replication. All right, there's 2 different ways that viruses are going to make more of themselves. And the main thing to remember is that they cannot do this alone. They cannot do this out in the environment. They have to be in a living cell in order to make more of themselves. Okay, so they're going to be dependent on the host's organelles, if this is a animal cell or a plant cell. Of course, in bacteria, there's not going to be organelles, but they're going to take advantage of those bacterial enzymes in order to make new copies of itself. And when they are using this lytic replication that we're going to talk about first, that's usually going to kill the cell sooner rather than later. There is a second kind called lysogenic replication that won't kill the cell right away. It probably eventually will, but the cell can maybe live on for maybe many generations till it actually dies. All right, so the 5 stages that we're going to look at here of lytic replication are listed here. Attachment of the virion to the host cell, how it gets into the host cell, synthesis. What I mean by that is synthesis of viral proteins as well as viral nucleic acid. And then how the parts of the new virion will come together and how it leaves the host cell again. All right, so those are the 5 steps of lytic replication. All right, so here's a figure from your book, lytic replication, and they use an example, these T4 bacteriophages. So remember, we're talking specifically now about bacteria that viruses that infect bacteria cells. All right, so a bacteriophage is a virus that literally eats bacteria, phage meaning eater. Okay, so bacteriophage only infects bacteria, and this particular one, T4, a bacteriophage, is specific for E. coli. Okay, so it will only infect E. coli. It's kind of astonishing, really, how specific it is. There's some bacteriophages that only will infect certain strains of E. coli or of certain bacteria. Okay, and this is a virulent phage. And what that term means is that it only does lytic replication. Okay, we're going to talk about lysogenic replication in a minute. And if you have a virulent phage, it will only do this replication cycle. Okay. All right, so first step here that it talks about, and I'm going to kind of break down this into so you can see it just a little bit better. First step is attachment. All right, so this figure is just labeled a little bit easier. You can see it a little bit easier. Okay, so number one, attachment. First thing to understand is that these bacteriophages, they are not moving in any kind of specific way. They are not responding to any stimulus. They're not, they don't have flagella so that they can move purposefully in any way. They are just moving around their medium in a random way. And if they happen to come in contact with an E. coli, then that's what they're able to attach to. So there's actually proteins on the end of these tail fibers that will recognize specific proteins in the outer membrane of the E. coli. Okay, so if they land there just randomly, then they will attach. And then they need to be able to inject their nucleic acid into the host cell. And it's easy when you look at this picture to think that, oh, this is just a big syringe, that tail spindle just kind of pokes down into the E. coli. It actually needs a little bit more help than that, because this peptidoglycan layer, even though it's thin in gram negatives, it's still a pretty rigid layer. And that's a pretty good barrier for anything to be kind of injected into a cell. So actually, the bacteriophage carries an enzyme along with it called lysozyme. And what lysozyme does, it breaks the bonds between the N-acetylmuramic acid and N-acetylglucosamine in peptidoglycan. All right, so it breaks the bonds between those sugars that make up peptidoglycan and enables the virus to, again, break through the peptidoglycan and enter into the cell. All right, so it facilitates entry into the cell. All right, so kind of back to our big picture here. Once outside of the cell, those tail fibers and that capsid, they just fall away. They're not necessary anymore. And now, so it's called a virion when that whole capsid and everything is together. Once it's inside the cell, then we would more correctly call it a virus. Now that virus is going to release enzymes that are going to degrade the host DNA, chop it up, basically. So that phage DNA is taking over the ribosomes to make its own proteins and using the enzymes of the cell. Remember, this is a double-stranded DNA virus to replicate more, synthesize its own DNA, and transcribe and translate its own proteins. Okay, so you see the phage proteins here. Again, all this is happening during what we would describe as the synthesis step. And then those proteins that the virus makes are going to assemble. So here's kind of a blow up of assembly. Kind of looks like a assembly line, a GM or something like that. We think that all these parts, we don't totally understand how they all come together right. We think that they pretty much just spontaneously do this. There may be some enzymes that are involved in making that all happen correctly. But they come together into mature virions, and then those mature virions are released. Again, we've got an intact cell wall in that cell, so it needs a little bit of help. Again, lysozyme is actually made during the synthesis phase, and that helps lyse the cell so that all those new virions can break out and go and infect other cells. Okay, so that in a nutshell is lytic replication. I like this picture here that your book, I think, no, that wasn't in your book. But I came upon that picture, and you got to just almost feel sorry for that E. coli, right? It's bursting open, usually on the order of about maybe 200 bacteriophage are going to be released from that blown open cell. So if we look at if we would think about constructing, we talked about growth curves for bacterial growth. If we did that for a virus, so if we had, one virus enter a cell, okay, in the medium, then it's in the cell, we don't see any during that synthesis and assembly. And then at some burst time, we get a whole bunch of viruses, again, maybe about 200 coming out of the cell all at once. So our growth curve is going to look a little bit different than what it would for bacteria when you have kind of a short lag and then exponential growth. Okay, the time here, about 25 minutes from entry into the cell till a bunch of them leave. So this is happening pretty quickly in viruses that infect bacteria. All right, so the other kind of replication that some viruses can do is called the lysogenic replication cycle. All right, and we're going to use as an example here lambda phage, and you can see that looks kind of similar to the T4, but it doesn't have the tail spindles. It's just kind of a stalk, and then that capsid that has is another double-stranded DNA bacteriophage. It's considered a temperate bacteriophage. That just means it can do the lysogenic replication cycle or the lytic replication. Okay, so a virulent phage can only do the lytic replication, and a temperate phage can do both. All right, so what is different about lysogenic replication? Well, it starts out the same, where by random motion, it's going to attach, recognize specific proteins there, and enter. So again, it's releasing lysozyme there so it can get into the cell. But what's different now is that instead of that virus immediately breaking up that host chromosome, it's actually going to integrate into the host chromosome. And we call that stage the prophage, okay, in the chromosomes. And it actually secretes an enzyme, I think, so that no other bacteriophages are going to infect that same cell. Because if you think about it, if you had a bunch of lambda all doing the same thing at once and all integrating into the chromosome, that chromosome is just going to be all full of holes, and the cell is probably going to die. All right, so just one at a time. It has a mechanism, so only one virus infected at a time. Okay, and I wrote that here, viral DNA incorporates into the host DNA. And that can just stay there for generations and generations of the cell. So we've got it dividing. Every time the cell divides, that viral DNA is going to be part of the host. Okay, so if you remember last time, we talked about one of our methods of horizontal gene transfer was transduction, where a little bit of viral DNA, bacterial cell DNA actually gets packaged up into a virion, and then a virus will take it to another cell. So you can see how that could actually give the cell some new properties. If it's not only if it's bringing viral DNA, but if it's bringing DNA from another bacterial cell. So it's, again, doing something to potentially change the DNA of that bacterial cell, and it doesn't kill the cell right away. It can live like that over generations. Question? Okay, good question. When would it go through the lytic cycle and when would it go through the lysogenic cycle? It depends on the virus, but hold that thought. Typically, when they go through the lysogenic cycle, eventually they will go through the lytic cycle. Okay, so I'm getting to that. Okay, so we think that eventually, and we don't know exactly what might spur the cell to do this, but eventually that viral DNA will come out of the host DNA, and we call that induction. Okay, in other words, let me write that here. Viral DNA leaves the host chromosome. Again, and we call that induction. Why does it do that? Sometimes we think it will do that if the virus can sense that the host cell is going to die. We know that we can make induction happen if we expose the cell to radiation. So we think maybe if that cell is in trouble, that will spur on induction. And when the viral DNA comes out of the host DNA, it will enter back into the lytic cycle. Okay, so it will break down that host chromosome, start to synthesize its own DNA, its own viral proteins. They'll assemble and lyse the cell and release. So really, lysogeny is just kind of an extension of the lytic cycle. So if a host does lysogeny, it's always going to be able to do the lytic cycle as well. Okay, and one other thing that I want to mention is that while that viral DNA is nestled into that host chromosome, it can actually enable the host cell to maybe have different traits than what it had before. And we call this lysogenic conversion. Okay. In other words, the bacteriophage can carry genes that can actually change the phenotype of the host cell. And when we're talking about phenotype of bacteria, we're thinking about things like, nutrients that it can now utilize, or maybe a toxin that it can now make. Some examples of when we know this happens, the bacteria that causes diphtheria, it only makes diphtheria toxin if the cell has been infected with a particular kind of bacteriophage. And that bacteriophage gives that cell the ability to make diphtheria toxin. The organism that causes strep throat, strep pyogenes, it can cause a very serious form of strep throat known as scarlet fever if it has been infected with a bacteriophage that enables the cell to give us this bright red rash. So there are cases, they're kind of rare, where we know that organisms have been able to kind of do something in addition, just because they've got instructions from a virus to be able to do that. Okay. All right, so we've just been talking about bacteria up to this point and viruses that infect bacteria. There are some differences now in viruses that are able to replicate in animal cells, right? So I call us animals. You could call it humans if you want. They've got the same basic replication pathway as bacteriophages. So, I mean, we don't have a whole ton of differences to go through here, but there are differences because a lot of viruses that infect animals are going to have an envelope. So if you think about it, if it's going to pull that envelope from the host cell, we can't have the cell just lysing and a bunch of viruses bursting out of the cell. It's not going to work. They're not going to be able to get their envelope that way. So some differences about how not only viruses enter the cell, but how they leave the cell, because again, they most often need an envelope. I think there is maybe one bacteriophage that's enveloped, and I don't really understand how that works exactly, but for the most part, bacteriophages do not have envelopes. Animal cells are eukaryotic. They have a nucleus. So viruses have to go to the spot in the cell where the enzymes are that it needs. So if it needs that enzyme to replicate its DNA polymerase 3 has to go to the nucleus, because that's where that enzyme is. And of course, animal cells lack cell wall. Okay, so just again, some differences about how they enter and how they leave again. All right, so 3 different mechanisms of entry of animal viruses. And I caught a little error here. This they fixed this on the new edition of the book of this figure, but they were talking about animal viruses, and here they call the virus a phage. So let's say virus genome instead of phage genome here. Cross that out. Again, the new edition has that fixed. 3 different mechanisms. So direct penetration, membrane fusion, and endocytosis. Okay, a virus that does direct penetration, this is typically going to be a virus that does not have an envelope. All right, because you've got the capsid here. These little red spots that's drawn in, that's just signifying, hey, there's some specific receptor that there's proteins on the outside of the capsid here that recognizes those specific receptors, and that's why it docks there. A pore opens up and the viral genome is injected inside of the cell. Okay, so we just call that direct penetration. Again, the capsid falls off. It's not necessary anymore. The second way is membrane fusion. So this is going to be enveloped viruses only. Okay, so here we've got a virus with, you can't even really see the capsid very well in that picture, but it's got this envelope around it. That the membrane of that envelope fuses here in step 2 with the plasma membrane of, say, one of our cells. And those viral glycoproteins in the envelope that were specific for whatever cell that was just fuses with the outside of the plasma membrane. The capsid falls off or uncoats, and then you're left with the viral genome. Okay, and the third way, we think this is probably the most popular way, and I'll try to explain why in a minute. This can be naked viruses or enveloped viruses. where basically the virus tricks one of our cells into enveloping it, okay, by endocytosis. So we talked about only cells that don't have a cell wall are able to do this. They extend pseudopods and just kind of enclose whatever that particle is into a membrane. Once the whole virion is inside of the cell, the envelope comes off, the capsid comes off, and you're left with the viral genome. And this could be a naked virus that does this as well. Again, it's just either way, naked or enveloped. We think that this is probably the most popular because you'll notice that it doesn't leave any evidence on the outside of the cell that that cell has been infected. Okay, over here, if the membrane of the envelope of the virus is left on the surface of the cell, there's kind of probably be some proteins on there that are specific to viruses. So that's kind of an advertisement for our immune system. Hey, there's something in this cell that is foreign. Okay, if there's some kind of spike protein there, for example, or anything, again, that's going to be a tip-off for our immune system. So we think more likely with viruses that infect us, more often than not, they're going to try to enter by endocytosis. Okay, so once that virus is inside of the cell, its prime objective not only is to make viral proteins, but it's going to have to copy its nucleic acid. So we talked about there's different kinds of nucleic acid. Talking a little bit more specifically here about single-stranded RNA. There's two different kinds of single-stranded RNA. We can have a plus sense or a minus sense. And I mentioned this a little bit last week, but plus sense RNA, this acts as messenger RNA. Okay, so that's the RNA that's read at the ribosomes. The negative sense RNA, this is the complementary strand that would match up with the positive sense. Okay, so it has to be copied. Again, the complementary base pairs in order to be read at the ribosomes. All right. So, you know, when we were talking about, that terminology of the DNA, like this is a template strand and this is the coding strand and the plus sense and the minus sense, I was trying to kind of help you understand, again, the difference between plus sense RNA and minus sense RNA. Okay, and the next graphic will help illustrate that a little bit as well. DNA viruses are usually going to have to get into the nuclear membrane to the nucleus, because again, that's where the DNA polymerase 3 is, that they need to replicate their DNA. RNA viruses usually don't have to go in there because the enzymes that they need are just going to be, they don't need RNA polymerase 3. They probably have to make their own enzymes to replicate their RNA. So they can just stay in the cytoplasm. And there is an exception to that, but that's kind of a general rule. Okay, so if you look at a little bit more closely, thinking about what's the difference between a plus sense and minus sense RNA, and what do they have to go through to make a usable form that's going to be read at the ribosome and to make more of themselves. If you have a positive sense RNA virus, good example of that, coronavirus, that's a plus sense RNA virus. So it comes into the cell, and then really handy that you can use that exactly to be translated into proteins at the ribosome, into its own proteins. To make more of itself, it needs an enzyme to make the complementary negative sense strand, and then that strand will be used to make more of the positive sense. Okay, so you can't make a positive from a positive, you have to go through a negative and then to get, okay, so in other words, you got to have the complementary bases here to get back to what you started from. Okay, a negative single-stranded RNA, influenza virus is an example there. You're going to get your complementary strand there, and from there, you can use that plus sense again to make its own proteins, and to use that to make more negative sense RNA. There's only one, I think, double-stranded RNA virus that infects humans, but basically that double-stranded RNA is going to unwind, and then complementary copies made of each of those to make more of itself, and some of that single-stranded to assemble into viral proteins. Okay, so the point is of this, no matter what kind of virus you have, and again, don't memorize the examples here, you know, whether you have a double-stranded DNA or plus sense RNA or minus sense, the virus has to have a way to get positive sense RNA, because that's the only thing that can get read at the ribosomes. Okay. And again, some viruses have crazy ways to do that, like HIV retroviruses have positive sense RNA, which they can just easily use to be read at ribosomes, but instead they go through this DNA intermediate. DNA is a little bit more stable, but again, they go through this whole rigmarole when it seems like it could be a lot easier. Okay, so once the synthesis step is done, you know, more nucleic acid is made, the virus is ready to come together and leave our cells. And the whole process of replication typically even if it's doing the Lytic cycle, typically takes a lot longer in viruses that infect our cells, on the order of 24 hours. Remember, we said the bacteriophage takes about 24 minutes, 25 minutes. So typically takes a lot longer. A couple different ways that non-enveloped viruses can be released from the cells. Either they are just lysed, either they're lysing the cells in the way that bacteriophage usually do, or they're exocytosed, meaning, and your book doesn't have a good graphic of this, but meaning they're just kind of escorted out in a membrane and then released. Envelope viruses have to be released a little bit more slowly because they need to get a membrane from somewhere. They can also do exocytosis, and especially they're going to do that if they actually took some of their membrane from the nuclear membrane and then sort of escorted the rest of the way out of the cell. Or the other mechanism here is by budding. Okay, so let me just make the point that exocytosis does not acquire a membrane. Again, it's just sort of escorted out of the cell without getting part of the cell's membrane on the way. Okay, your book does have a nice graphic here showing budding. So in budding, that's when the virus, the new virus, is going to acquire some of an envelope from our plasma membrane. Okay, so the virus is going to express some proteins right in its host cell membrane. So that's what these little spikes are. And then on its way out, it's going to grab some of that host cell membrane and leave with that. Okay, so that, again, that's a method of virus leaving that we refer to as budding. So if you think about it, again, like I said, that has to happen a little bit more slowly because, or it typically does. So typically, if you again look at a growth curve of a membrane-bound virus, it's typically going to be leaving the cell a little bit more slowly. Okay, over here first question.
On the slide above, the glycoproteins that it has, like when it exits, are those there from when it enters, or were those like?
Okay, so when did these glycoproteins get there? My understanding that happens during the synthesis phase. So the virus is, making more of its, say, DNA or RNA and more of its own proteins. So it will make those glycoproteins and they will get expressed on the surface of the cell. Okay, good question. And over here, Lee?
Through budding, it gets like the envelope.
Yes, yes. So through budding, that's how it acquires its envelope. Yep. Okay. Now, sometimes virions will leave a host cell so quickly that it effectually lyses the cell, because a host cell can only stand so much of its membrane being disrupted before it's not going to be viable anymore. So again, sometimes that happens really quickly, and a bunch of, again, that can kill the host cell pretty quickly. But I was just comparing it here to, what the bacteriophage and how they tend to kind of burst all at once. Okay, so do viruses that infect animals, do they infect, what kind of replication cycle do they do? Do they do lytic? Do they do lysogenic? They do both, but we call the lysogenic cycle, when it happens in an animal host, we call that latency. Okay, so this is basically the lysogenic replication in animal cells. All right, it's the same thing, we just call it something different when it's in animal cells. Okay, so when animal viruses remain dormant in host cells, okay, and just like with bacterial cells, that can remain dormant for really a long time. Viruses can be dormant in our cells for years and years. Some of them actually don't integrate right into the host cell chromosome. Some of them form little tiny circle plasmid type structures. Whether they are right into the chromosome or they make plasmids, that viral nucleic acid gets replicated every time our cell divides. Okay, so that can last for years and years. Herpes virus is one that can go dormant, and often does go dormant. Years later, that can reactivate. If you have chickenpox as a child, that's a herpes virus that can reactivate, usually later in life, in an infection known as shingles. Okay, so that's one example. There's not too many that can go latent. HIV is another one, but we think that this is always permanent. So once that herpes virus or HIV integrates into our chromosome, that it doesn't ever come out again. Not like in bacteria where you can have induction. Okay, so we just did that, so I'm not going to go through that again. But so again, just a little bit of, we have lysogeny and bacteriophage. We call that latency in animals. In bacteriophage, you get a prophage. And remember, that's the step right here where the virus is integrated into the chromosome. We don't call it a prophage for animal hosts. We call it a provirus stage because it's not a phage. But again, either it's going to integrate right into that chromosome, and of course, our chromosomes are not circular. but sometimes that virus will form these little plasmid-like structures. We call them episomes. Herpes virus does that. And for bacteriophage, right, they can come out of the host cell chromosome and, you know, start breaking up the host cell chromosome. We call that induction. For animal cells, there's no induction. We think once that nucleic acid is mixed in with ours, that it doesn't ever come out again. So that's why we think there's a really good chance that HIV, we won't ever have a cure for that, because once it's integrated into our chromosomes, we don't have any way to get it out. The best we can do is manage the symptoms, which we've gotten pretty good at now with lots of drugs, but we'll talk more about that later. Okay, all right, we're doing okay for time. I want to talk a little bit briefly on how do we grow up viruses in the lab. They have to be in some kind of living cell. All right, so we can grow viruses in bacteria. We can grow them in larger organisms as well. But if we grow them in bacteria, and it's been a long time since I've done this, but typically you've got your growth medium, say in your petri dish, you put a lawn of bacteria, meaning you inoculate that really heavy with your bacteria of choice, you grow that up, and then you add your suspension that's got the viruses in it. So say you want to know, about how many viruses is in this suspension, and you have to have a species of virus that's specific for the bacteria cells that you put on there, but you incubate it, and then wherever there was a bacteriophage, it will cause the lysis of the bacteria cells that it infects. And then those cells will lyse the other cells, which will lyse the other cells. So basically you get these little plaques formed wherever there was a bacteriophage. Okay, so you could extrapolate back by counting the plaques, how many phage were in your original sample. So it's sort of the opposite of the viable plate count. So if you were in lab, and we talked about that in here, where you're counting the number of colonies for the viable plate counts, here you're counting where the bacteria have disappeared, essentially, because they've been lysed. You can also grow viruses in eggs. And this is a really common way that we grow up enough flu virus, so that when we deactivate that and make that into flu vaccine. And you can actually, depending on the virus, inject it into these fertilized eggs, inject it into lots of different parts of the egg. The yolk sac is a really, really nutrient rich place, so we can grow lots and lots of viruses this way. Okay, and that's still the way most flu vaccine is prepared. The other way that we can grow viruses is in a cell culture. We used to call this tissue culture, but we recognize, well, tissue usually means a couple different kinds of cells, and that's typically not what we're doing. We're growing a single kind of cell. So we more often call this cell culture now. And sometimes you'll do this in some kind of a plastic well dish, where you'll have a monolayer of cells growing. It's bathed in some liquid that's super nutrient rich. And then your viruses infect those cells. The really difficult thing with cell culture has always been trying to keep bacteria from contaminating it, because you've got all these nutrients, so you have to add antibiotics in there to try to keep bacteria from contaminating it. In something like this setup here, I can see the advantages of this because it's not open to the air. So you've got your cells in there and your medium that's feeding those cells, and then you kind of inject in your viruses, and it looks like maybe you can take out old media and add new media, maybe to kind of keep those cells going. Using cells is going to be a lot less expensive than using fertilized eggs. Take up a lot less space. There's two different kinds of cells that are commonly used, either diploid cells or cells that can be continuous. Diploid cells, these are typically stem cells that from either plants or animals that are going to grow for about 100 generations and then they die. That's just, you have a normal kind of lifespan of a cell line. But if you use cells that are derived from a tumor, basically that's what's special about cancer cells. They just grow and grow and grow and grow, and there's no end to them. So one particular kind of tumor cell that's been commonly used in cell culture are HeLa cells. That's kind of the most famous one. They were derived from a woman, Henrietta Lacks, that died of cervical cancer in 1950s. So they used actually some of her cells and have been, perpetuating that cell line ever since. The problem with these continuous cultures is that you get mutations that kind of build up, and some of those cells actually aren't even diploid anymore because whole chromosomes have kind of been deleted due to mutations. So there are some issues with that, but okay. So lastly, we talked about kind of, hey, these are all the basic things about viruses. And I still want to talk about prions as well. I think we have time for that. So this is probably will be kind of a debate that won't ever go away. There are scientists that kind of debate one way and, knowing scientists that debate the other way. But if you think about what are the traits of a living organism, really 4 traits. So living organisms are able to grow. So when you think about, do viruses grow? Viruses, no, not really. You don't have baby viruses that grow into adult viruses. They're just assembled. Okay, so we have to say probably no, they don't grow. Do viruses reproduce? Sure, they do, but they have to be in a living cell. Now, there are some bacteria that also have to be in a living cell in order to reproduce. So that by itself is not a deal breaker that they have to be in another cell. Are they responsive? And that means, do they respond to stimuli? Do they act, can they kind of change their behavior in response to a stimuli? That's the one thing that people argue about, because you could argue, yes, viruses evolve over time. What was the other thing? And they do evade host defenses to some extent, but whether you consider that a real response or not, in general, the answer is no. But again, some people argue about that. Do they have a metabolism? Definitely not. No. Okay, so most people would call viruses a particle and not call it an organism, because they, again, definitely for the most part don't meet those requirements. But again, people argue about that. Okay, so I want to finish up our time here in chapter 13, thinking about things smaller than a bacteria cell, and talk about prions. Prions were, we knew that there was some kind of infectious particle for quite some time, but these were named in 1982, so seems like yesterday, by Stanley Prusner. And he gave these particles the name prion. He called them proteinaceous infective particles. So when you kind of squish those 3 words together, he came out with prions. And what is really mind-blowing about these particles, okay, they're proteins that cause disease. Okay, so they have no nucleic acid, no DNA, no RNA, and yet they could reproduce themselves. So this was a really mind-blowing thing. For a while, people called them slow viruses because they seem to take, a long time to reproduce. But we now know that they are just proteins. So how does that work? Well, the protein in question here, we call prion protein. So we abbreviate that PRP. So this is a protein that we know that's in all mammalian cells. It's associated with the cell membrane, and we don't really understand what its real function is. But we know that everybody has it. And apparently this protein can become misfolded. So here's the normal cellular PRP and what that protein looks like normally. And when it becomes misfolded, how is that different? Well, it's got some more beta sheets, apparently. It folds different, so it doesn't work the same. So when does it become misfolded? Well, when it's exposed to a misfolded one. And we'll talk about there's other ways. Sometimes this can spontaneously happen or genetically happen. But if that protein is exposed to a misfolded one, then it will cause the normal ones to become misfolded. So, one bad apple and a bushel of good ones will ruin the rest of the apples. All right, you maybe heard that expression before. Okay, so what's the so what if this prion PRP prion protein gets misfolded? These are going to kind of build up. They're not going to function, and eventually the neurons are not going to be functional. Okay, and this can cause basically some plaques in the brain. Okay, so if you look at this picture here from your book, comparing normal brain tissue on the left and brain tissue from, I assume that it's an animal that's got prion disease, you see these kind of plaques here, and that's where the name spongiform and encephalopathy came from. Kind of looks like, those big holes in a sponge. All right, so what are some examples of prion disease, and how do we think that prion disease is passed between animals? Probably the most famous one is bovine spongiform encephalopathy. Oh, should have practiced saying that. Otherwise known as mad cow. All right, so that came on the scene in the late 1980s, early 1990s, especially in Britain, where you had these cows kind of staggering around and dying. We've soon figured out that we had to stop giving animals feed that had dead animals ground up into the feed. So we found out that that's how it was propagating. When people ate cows that had died of mad cow disease, then that disease was called variant Creutzfeldt-Jakob. And this was in the news all the time in the early 1990s. Only about 200 people died of it until we figured out that what was propagating it in animals. And I was in England in 1991, and everyone said, "Don't eat hamburger, don't eat beef." I said, "Well, what's the big deal? We cook it." Well, to deactivate prions, you have to cook meat at almost 500 degrees Celsius and keep that temperature for 4 hours. So just cooking it on the grill is not good enough to deactivate the prions. There is a form of Creutzfeldt-Jakob that is just genetic. Okay, so you could get it by either, eating contaminated meat, or there is a kind of rare genetic form of that. Scrapie is another prion disease that sheep would get. It would cause them to act really oddly. They would like scrape up against posts, so their wool was gone and their skin was all raw. And chronic wasting disease, okay, so that's sheep, that's deer in Michigan have been found to have chronic wasting disease, elk and moose. It just causes them to kind of waste away. So how do they pass it from each other? We think bodily fluids maybe, or just being in close contact with each other. So far, there haven't been any cases of anybody eating venison and getting prion disease, but of course, that's a real worry. All these diseases, all prion diseases, they're fatal. So it might take, a number of months to kind of play out, but they all have neurological consequences and they're always going to be fatal. So how do people get it? Either it's inherited, meaning you have some kind of rare, very rare genetic mutation. There's another prion disease called fatal familial insomnia that runs in families. It can be sporadic, meaning the gene has not mutated, but then there's this kind of rare misfolding event that happens. Again, that's another way that people can get Creutzfeldt-Jakob, or infectious. So they eat some meat that's been contaminated. There was another prion disease called Kuru that was from a tribe in New Guinea. You might have heard about that. It was their culture to, in order to pay respect to a relative that died, they would eat that relative. And it was, especially an honor to be able to eat their brain. So they were passing this prion disease because they were eating the brain tissue of people that have died of prion disease. So finally we're taught not to do that anymore. And so that Kuru has died out. But so anyway, really lots of fascinating things to do with prion disease, but just kind of hit the highlights there. So exam Tuesday, we'll move on to chapter 9 on Thursday. I will be in my office until about 4 o'clock today. So anybody still wants to look over the last exam or you have any questions? Okay, otherwise have a good weekend.